California. I'm pleased to make a presentation on this occasion to the Moldovan Academy. Of course, I regret that I'm not able to be with you in person. This was my plan, but life has intervened, as you know, and I'm not able to travel. But I hope that this presentation will give you a flavor of the work that we've done in my laboratory and the nature of the role of basic science in the advance of medicine and biological science. Let me begin with some basics, and that is, first, the nature of genetic information is contained in the DNA molecule, which is depicted on my next slide. As I'm sure many of you are aware, the genes that control all of the processes in the cells of our body are housed on chromosomes in the nucleus of a cell. Each gene of the 23,000 different genes that are in human chromosomes encodes the information for a protein molecule. Proteins are basically long threads of molecules composed of amino acids that are stitched one next to another. And after they are made, these very long stretches, sometimes over 1,000 amino acids in length, fold into a compact three-dimensional shape that gives each protein molecule its unique structural and functional qualities. Now, one very basic tenet of modern biology is referred to as the central dogma. This dogma, which was first considered by the pioneers in this field, James Watson, Francis Crick, and others, holds that the DNA molecule contains the blueprint, but that blueprint must be read. And the way it's read is by copying DNA into another molecule, a variant of DNA called RNA. A single strand of RNA is copied from genes in the nucleus, and that strand of RNA is transported into the cytoplasm of the cell sap, where it is recognized by a little machine, kind of like a sewing machine, called a ribosome. The ribosome then reads the information in the RNA and uses that information to place amino acids one next to another, where the ribosome basically staples these amino acids together, and that then allows the protein molecule to be made, and as I indicated previously, to fold in this compact shape. Now, DNA is universal in the world, at least on Earth, and it even is found in microorganisms, viruses, such as you see here. This is a cartoon of a virus that infects not a human, but a bacterial cell. It sits on the surface of the bacterial cell, and it injects its chromosome into the interior of the bacterium, where the bacterium then can be taken over by the machinery encoded by the virus. Viruses aren't alive themselves. They reveal themselves through infection of a host cell, in this case, a bacterium. Now, of course, the bacteria are not powerless. Through evolution, bacteria have learned to try to evade these viruses by creating protein molecules, enzymes, that recognize the viral DNA as foreign and then break the viral DNA into pieces, thus preventing the virus from being infective. Now, I bring this up because this is the source of the modern revolution called genome editing that has given rise to a technology that you may have heard of called CRISPR or CRISPR-Cas9, where the enzymes that are made by the bacterium using both a protein and an RNA molecule can be engineered to make precise incisions, indeed corrections, at sites in even the human genome. This is the basis of the recombinant technique called gene editing. Now, I have a little film from one of the founders of this field, a colleague of mine, Jennifer Doudna at UC Berkeley, And I'd like to share that with you. That explains very briefly the technology behind CRISPR-Cas9.
Scientists have long imagined being able to make specific changes to the genetic code of a cell or organism. A revolutionary genome editing technology invented by scientists at UC Berkeley and Umea University makes this possible using an RNA-guided protein called Cas9. Cas9 searches through the cell's vast genetic material until it finds a sequence of DNA that matches the sequence of its programmable guide RNA. The guide RNA enables Cas9 to open the DNA helix and to position two molecular blades to cut each strand of the DNA double helix. Repair enzymes seal a gap in the broken, sometimes by inserting new genetic information supplied by the cell or the experimenter. In this way, disease-causing mutations can be corrected by changing the underlying genetic code. Scientists are now using Cas9 technology for research applications in human health, agriculture, and bioenergy.
Now, of course, this technology offers great promise, not only for creating the next generation of drugs, but also for possibly correcting human genetic disorders, diseases such as sickle cell anemia, or certain forms of cancer, or certain forms of dementia, such as an Alzheimer's disease, where the disease results from a single mutation. There is the prospect that these could be changed, these mutations could be changed in ways that would prevent the illness from onset. Now, of course, the danger and the concern is that by using this technology, we might be have freedom to change other things that may have more severe societal implications. For instance, what if we could change the personality of a child? We could change the fate of a child by creating a higher level of intelligence, or the more perfect athlete, or the more perfect soldier, by changing the germline, that is the embryonic DNA of a baby, with certain engineered mutations. This is of great concern, and the world community has now tried to halt all further work on changing the genome in an embryo because of the potential misapplication of this technology. So it has great promise, and there are very serious ethical concerns with the application of this technology that you may and really should be aware of. Now, I'd like to turn for the next few minutes to a description of my work, and I'll start with a history of the subject of biological membranes. For many years in my laboratory at the University of California, Berkeley, my students and I have studied how this structure, which is a cartoon of a biological membrane, is assembled in a living cell. Let me illustrate the points of this cartoon. These red structures are called lipids. Specifically, they're called phospholipids. Phospholipids have a surface, these red spheres, that is, these are molecules that are polar. They love water. They face both sides of a membrane. This side may face the outside of a cell. This surface may face the inside of the cell. And the interior of a membrane consists of these long strings, red strings in this case, that are apolar. They're fatty acids. They are covalently attached to the polar or hydrophilic head groups. And these phospholipid molecules spontaneously arrange themselves in a bilayer so as to favor the apolar interactions in the interior of the membrane and the polar surfaces that surround a cell or surround organelles, little membranes inside of a cell. Now, by itself, these bilayers are completely impervious to charged molecules. A membrane that had only these phospholipids would not be able to selectively acquire ions, sugars, amino acids, things that cells need, or toxins, things that cells need to get rid of. And so for that reason, membranes have evolved to contain membrane proteins that provide the individual personality of a cell. For instance, a green structure such as you see here may be a channel, a very selective channel, through which small molecules may pass in one direction or the other, or other molecules may represent receptors, kind of like antennae on the surface of a cell, to which hormones, molecules like insulin, bind to trigger the cell to produce some desired biological response in the form of a metabolic pathway. So there are literally thousands of membrane proteins, some of which populate a membrane that surrounds the cell, that contains the cell and allows the cell to communicate with its environment, and others, as you'll see in a moment, that are interior to the cell and surround specialized compartments that allow the cell to survive. So this is the biological membrane. Now let me show you an example of a membrane of a very important cell that we all rely on. This is a cell in our pancreas called the beta cell. This is a cell that's specialized to produce the hormone insulin. Insulin, as you know, is a molecule in our blood that recognizes, as we have a meal, the accumulation of glucose in the bloodstream. And the insulin molecule is produced when the glucose levels rise, and it travels around in the body, binding to receptors on appropriate cells, triggering these cells like fat cells or muscle cells to swallow up glucose through specialized glucose channels. The glucose is then made into a polymer, a storage polymer called glycogen, which is later used when we exercise or as we need to derive energy from the glucose, and the cells then break it down for its further use. So insulin is a very key hormone. It was discovered in Canada in 1920, and as you know, it is the major deficit in patients with juvenile diabetes. We know a great deal about how insulin is made in a cell, and I want to tell you about one of the great founders of modern cell biology, George Palade. George Palade was the first to really harness the power of electron microscopy to visualize the internal structure of cells, particularly cells in experimental animals, but the same cells we have in our bodies. And he was able to show that one of the steps in the secretion of insulin is its capture into a membrane-enclosed granule, such as you see here. Many such granules house insulin, allowing it to be expelled at the cell surface. A granule migrates to the cell surface and touches the membrane surrounding the cell, and then expels the internal particle of insulin, which then dissolves and is eventually distributed into the bloodstream. Here's a picture of Dr. Palade later in his career when he was a professor at the University of California, San Diego, seated behind his favorite instrumental tool, this electron microscope, which projects a beam of electrons down this column, bombarding the sample placed in this chamber, and the scatter of electrons can be visualized in the form of a shadow image of a thin slice through a cell. He and his colleagues devised the procedures that allow these thin sections to be preserved against the attack, the bombardment by electrons. As you know, Dr. Palade was a Romanian scientist of enormous intellectual depth. He won the Nobel Prize in 1974 for his discoveries using this electron microscope, having done most of his work in New York at the Rockefeller University. Indeed, in the 1950s, Palade was the first, using the electron microscope, to visualize ribosomes in the cytoplasm of cells. These are, as I said, the machines that make protein molecules. Well, one of the other places where these granules that house molecules like insulin has a very important role is in nerve cells. This is an image of a nerve cell, in this case, the end of a nerve cell nearly touching a muscle cell. And nerve cells have these very small granules. They're called vesicles, in this case, synaptic vesicles. Synaptic vesicles don't have insulin, but they contain chemical neurotransmitters of the sort that control all of our thought and emotional processes. It is these neurotransmitters that guide the activation of nerve cells. And in this case, a nerve cell is responsible for stimulating muscle contraction. When these vesicles in a nerve cell at the neuromuscular junction, when these vesicles touch and expel chemical neurotransmitters out of the cell, they diffuse into this gap called a synapse. And on the other side, the chemical neurotransmitters bind to different receptor molecules that cause the muscle cell eventually to contract. In the brain, there are billions of nerve cells in the human brain. Each one may make thousands of connections to different nerve cells, such that these synaptic connections can be over a trillion in a human brain. And each one guides the processing and transfer of a bit of information within a neural network. So everything that we know and do, how we feel, and what we remember is guided by the pattern of fusion, of merging of these synaptic vesicles at the nerve terminal. Now, when Dr. Pallotti did his work in 1974, all of it was at this level of understanding how the cell, what the cell looks like, and how molecules may flow within the cell, as I'll describe in a moment. But I heard him speak about this work in 1974 when he had just returned from Stockholm to give a lecture at a cell biology meeting in San Diego. And what was striking about his discovery was that at the time, in 1974, not a single molecule was known to be involved in organizing this process. Surely there must be many genes that control this process, many gene products or proteins that allow vesicles to be made in the cell, that guide them to the right place on the surface of a cell, that allow them to merge with a membrane to promote this last step in secretion. And yet, in 1974, not a single molecule, not a single lipid, not a single protein molecule had been ascribed a role in this process. So when I began my career in 1976 as a young faculty member at the University of California, Berkeley, I decided to study this process in a simple microorganism, an organism that I believed would have the same process, the same pathway, but one where it was possible to use simple genetic and biochemical techniques to understand the nature, the mechanics, the mechanism of this process called protein secretion. So I chose as my experimental subject a very simple microorganism called yeast, or Saccharomyces cerevisiae, baker's yeast. Unlike bacteria, yeast cells are more like animal cells in that they have a nucleus, and you'll see an example of this in a bit. Yeast cells have a nucleus. They have their chromosomes in a membrane that surrounds the nucleus. But unlike animal cells, yeast cells, when they begin to grow and divide, don't divide in half. What they do is they send out a small bud. When the nutrient conditions are correct, the cell starts to grow and it sends out a small bud. And over the next 90 minutes or so, as the cell is growing and dividing, this bud gets bigger and bigger and bigger until it approximates the size of the mother cell, at which point the two cells divide in half, and the daughter, the bud and the mother cell can go on and repeat this process as the nutrient conditions permit. Now, it's clear from a simple inspection of this cluster of cells, such as you might see growing on the surface of a grape, that if this process can be visualized, it must involve the selective addition of new surface molecules, new membrane surface that is localized on the bud of the growing cell. What may allow this bud to grow in preference to the rest of the cell? Well, using the technique that Pallotti had pioneered, it's possible to look inside of a yeast cell by cutting a thin section through the cell that has been treated and embedded in plastic, allowing it to have its structures, its cellular structures preserved. doesn't look nearly as interesting or dramatic as a pancreatic beta cell, but the yeast cell nonetheless has many ribosomes. These darkly staining spots in the cytoplasm are ribosomes. This is a structure that is called the vacuole, similar to a human cell structure called the lysosome. There are threads of membranes generally up against the surface of the cell that are another membrane that I'll mention in a moment that's involved in the biosynthesis of proteins that are going to be exported. But our attention, when I began, was drawn to a cluster of small granules or vesicles, such as you saw a moment ago at the tip of a nerve cell. A yeast cell has the very same kinds of vesicles. Of course, these vesicles don't have insulin, or they certainly don't have chemical neurotransmitters, but they have instead the enzymes that must be exported to the cell exterior that allow a cell to grow and salvage carbons from its environment, such as it finds on the surface of a grape. Now, my first graduate student, Peter Novick, and I also speculated that these vesicles, having a biological membrane themselves, might constitute the building block for the growth of the plasma membrane, so that as these vesicles move to the cell perimeter and this membrane joins the membrane of the cell surface, each event, called a membrane fusion event, would permit the membrane of the cell surface to expand by one quantal unit, bit by bit, as hundreds or thousands of these vesicles merge with the surface membrane. The cell would enlarge, eventually to reach the size of the mother cell. Now, that very simple prediction has a powerful consequence, and that is, if this is true, then genes required for the manufacture of these vesicles, for their traffic to the bud, for their fusion by membrane fusion at the cell surface, each one of those genes would be essential. That is, if you crippled the gene by chemical mutation, by introducing mutations into the gene, the cell would die. Now, there are many such genes in yeast that are required for the cell to be viable. And we, in my laboratory, quickly developed a simple way of evaluating these cells when exposed to random mutation by a chemical mutagen, when these cells have a specific defect in the process of secretion. We asked, are there cells that, as they are dying, accumulate enzymes that would have been secreted outside of the cell, instead accumulate them inside the cell? Now, we had one such mutation. We called it SEC1, short for secretion 1, in 1978, in May of 1978. And just by chance, Dr. Pilati visited Berkeley for 2 honorific lectures on his pioneering work. And the graduate students in the department organized a dinner on his behalf. And Peter Novick, my student, described his initial results on this mutant to Dr. Pilati. And Pilati said, well, now, of course, you must inspect this cell by electron microscopy, which, of course, we were going to do. But we raced ahead to do this. And one of the great scientific memories of my life came that summer when Peter excitedly called me down to the basement of our building to show me the following image, which is of a cell as it's dying. The SEC1 mutant cell, as it's dying, now accumulates, instead of a small number of vesicles in the bud, accumulates many thousands of these vesicles, filling the entire cytoplasmic compartment. Indeed, the cell becomes choked with these vesicles, so much so that it dies because it cannot continue to build the surface as these vesicles fail in that last step of membrane fusion. Now, this was, of course, a very exciting discovery, and it then allowed us, using our procedures, to go forward to isolate many more such genes. And so over the next year and a half, Peter and my laboratory technician isolated over 200 more such mutations, defining a couple of dozen different genes that are required at different stages in a linear process of protein secretion. The pathway that we found is formally the same as the one charted by Pilati using other techniques. electron microscopy. But in our case, we were able to show that each stage in this process of movement of a protein from its site of synthesis through various membranes in the cell, finally to the formation of a secretory granule to the cell surface, each stage in this process required a number of different genes. And so we were able, with those genes, eventually to figure out what the molecular mechanism is that accompanies each of these stages. Now, as we were delving more deeply into the molecular mechanism, these genes from yeast were isolated by cloning, and they were sequenced, but the sequence didn't really tell us exactly what the protein does. But fortunately, the genomes of higher organisms, including that of humans, became available, and we discovered that the genes that we found in yeast have their formal equivalents in the mammalian genome. That is to say, this process of secretion has been conserved over 2 billion years that separate yeast cells and human cells. Now that has itself a practical implication, because if this is so, if the yeast and human processes are so similar, it should be possible, indeed it was, to take the gene for important human proteins of clinical value and get those genes to be expressed to produce their protein product in a yeast cell. In fact, I helped a biotechnology company engineer the introduction of the gene for human insulin into yeast. And now, many years later, a European country, Novo Nordisk, has the license to this product, and they manufacture in giant fermentation vats the production of 1/3 of the world's supply of human recombinant insulin. This company that I helped consult also was able to use yeast cells to manufacture a vaccine against hepatitis B. In fact, if you had such a vaccination, you have received little vesicles that are produced inside of a yeast cell. Now, I'd like to turn briefly to something that is actually related to the theme of membranes that I've been discussing, and that is, of course, the subject of great current concern, the COVID-19 pandemic caused by a virus called SARS-CoV-2. Here is an image of the virus. The reason I make the connection to the work that I've just described is that these viruses, the coronaviruses, are like little vesicles. They have a biological membrane, and their coat surface is a membrane protein called the spike protein. And this spike protein is the target of the 100 or so different efforts to create a vaccine in various places around the world. Now, unlike a vesicle such as a synaptic vesicle or a yeast secretory vesicle, these viruses have RNA, a nucleic acid, as their genome. The nucleic acid is housed inside of this membrane bilayer. Now, the virus infects cells through an interaction between the spike protein on its surface and a protein that has a normal function in a cell, a protein called ACE2. And this provides a portal, an entry for the virus into the cell. Once inside the cell, having bound to this receptor, the viral membrane fuses by the same process of membrane fusion that I've described to inject the RNA molecule into the cell interior, where the RNA is copied, and then the cell is subverted to make viral proteins by the cell's ribosome machinery. The objective now around the world is to create a vaccine, which is an antibody molecule that recognizes the spike protein and will thus prevent the virus from binding to its normal receptor. These antibodies that are produced have so far proven effective, at least in neutralizing the virus particle, but the question remains whether these neutralizing antibodies produced by a vaccine will be strong enough to prevent the infection process in a human. We'll know shortly if this is true. Now, last comment on coronavirus and SARS-CoV-2. This is an electron microscope image of what a cell looks like when it's been invaded by all these virus particles. You see them still on the cell surface, having just landed on its receptor. They become internalized into membranes, shown here, and occasionally the virus, as you see an example here, the virus touches the membrane of the intracellular membrane, and these two membranes merge, and the nucleic acid inside the virus is injected into the cytoplasm, where it then exploits the cellular machinery to make copies of itself. So there is an enormous basic and practical interest in how these viruses hijack the pathways that I've described in yeast and in human cells. Now I'd like to turn for my last few minutes to a discussion that will digress to some extent from the themes that I've already discussed. It represents yet another disease, a disease of growing concern that afflicts millions of people around the world, and with which I have a personal history that I'd like to tell you about. That is the disease called Parkinson's disease. Parkinson's is, in a very, very real sense, a pandemic. It's not infectious like coronavirus. We know of certain genetic origins of this disease, and yet there are many more patients who have no evidence of a genetic malady as the basis of the disease, who succumb sometimes after 30 or more years of illness. It is growing in prevalence as we age, as the population ages because of improved public health. We are experiencing quite a substantial growth. In fact, Parkinson's disease is the most rapidly growing neurodegenerative disease in the world. In the next decade or two, we will have approaching 20 million patients where there really can be very little, as you'll see, very little done. Thus, the need for more research in this topic. Much of it is attributable simply to aging. Some of it is to pollution and other genetic influences that I'll describe. The disease, like coronavirus, knows no geographic boundaries. It afflicts people all over the world. The growing fraction of these people are in China for reasons that aren't entirely clear. Within the next few years, over half of the patients who have this disease will be from China. Now, the problem with the disease, there are many problems, but one of the problems is simply in categorizing the disease. The disease presents itself with many different initial symptoms, as I'll illustrate. And the disease progresses at different rates, depending to some extent on those initial symptoms. There are forms of the disease, often they come later in life, that result in low survival, rapid dissemination of the disease, disabilities, including dementia that set in quickly sometimes. And on the other end, there are some forms of disease that may come on early in life. Sometimes patients as in their 20s are presented with Parkinson's disease. Sometimes these people can survive for decades, but with a very severe disability. The disability develops progressively over time, and yet sometimes it also does lead to dementia. It's not likely to be a single disease. It's more like a spectrum disorders, perhaps something like autism, with many different origins and many different paths of progression. And yet the pharmaceutical industry, having invested billions of dollars in the development of, or the attempts to develop effective treatments, must consider this as a single disease. And so as a result, most of the drugs, indeed all of the drugs that have been devised in recent decades that seem promising end up failing, not being generally applicable. There are really only two treatments that mitigate the symptoms. One is a replacement for the missing neurotransmitter dopamine that can be taken orally that restores normal movement for some years, but not forever. And a neurosurgical procedure where electrodes are implanted in the brain to stimulate an area that controls the awkward movements that often progress as the disease develops. These two treatments work, but they do not change the arc of the disease. The disease is inevitably fatal. Of course, there are many drugs that continue to be produced with which one is hopeful, but so far the problem is we don't understand where the disease begins. We don't understand any of the molecules that are implicated in the disease. There are no ways, there are no biological markers that can be used to follow the progression of the disease, as there is in other diseases like heart disease and cancer. So the frustration is that we don't have a basic understanding of where the disease originates. And here's a statement from one of the prominent scientists in this area, a biochemist in Scotland. There isn't a lot of funding, he says, to do the fundamental research on one gene or one protein that would be needed to really understand these things. Funding bodies want us to solve diseases and to work with companies to figure out shortcuts that can be made into a drug. But, and I emphasize this, we don't have the fundamental basis that is needed to really solve these problems. So how to attack the problem? I became involved in an effort to organize scientists around the world to study the basis of Parkinson's disease. And the reason I became involved was my wife was afflicted with the disease, suffered for 20 years progressively with dementia, and died several years ago. I was approached at that time by an organization called the Sergey Brin Family Foundation. Mr. Brin is the co-founder of Google, and as a result, of course, he has enormous resources. He also has a family history of Parkinson's disease. His mother has a genetic form of the disease, and he has inherited one bad gene. And so he is himself at some risk. He became interested in helping support efforts to understand the disease. He had funded an organization called the Michael J. Fox Foundation in the US, which is a wonderful organization that supports clinical research on Parkinson's. But Mr. Brin decided after some years that focusing entirely on the clinic was not going to lead to the basic discoveries that we needed to make. And so he created another substantial fund to help identify the molecular basis of the disease. And I was asked to become the scientific advisor, chief scientific officer of this fund. The fund is called Aligning Science Across Parkinson's Disease, ASAP. And our goal, which we have now launched, is to create teams, to identify teams of investigators around the world who are already engaged in basic studies, and some of which are already focused on Parkinson's disease. We have created a scientific strategy and a mechanism to direct a very substantial sum of funds to build an international consortium of investigators who are focused on trying to find the origins of the disease. The disease is kind of like a big puzzle. This is a simple puzzle, a cartoon that I've shown here, but the disease is much more complex. As in a puzzle, the disease has different pieces to it. One piece is the obvious pathology of nerve cell connections, the synaptic connections that I told you about that allow a brain to function properly. The disease has an effect on a protein that's inside the brain, a protein called alpha-synuclein. And in patients with Parkinson's disease, this protein builds up in aggregates in the cells of the brain that are making the chemical transmitter dopamine. These structures, these aggregates, are called Lewy bodies. They were discovered 100 years ago, and they are often a hallmark of the disease. Unfortunately, they cannot be detected until the patient dies, and a piece of the brain is taken by biopsy to be studied. Very often one sees these Lewy bodies in the region of the brain that makes dopamine, but also in patients with dementia, these Lewy bodies seem to be all over the brain. There are other pieces of the puzzle. A failure of the cells to remove protein aggregates like Lewy bodies that might be pathological and cause the demise of cells that make dopamine. Or there are other aspects of the disease that affect the energy powerhouse of the cell. the mitochondrion. In some cases, damaged mitochondria fail to be removed by the cell, and the accumulation of damage itself can be toxic to the cell. There is a process of cell death that eventually kills progressively all of the cells in the brain that make dopamine. This process could perhaps be controlled if the disease were to, if we were to find a way to prevent the death of these cells. And finally, there are aspects of the brain immune system and even the body's immune system that may initiate or make worse the degeneration of the region of the brain that makes dopamine. These are so-called neuroinflammatory processes that are poorly understood that may affect the progression of Parkinson's and of Alzheimer's disease. Well, finally then, let me tell you about the strategy that we have developed. Our team involved in starting this program has identified 4 areas for our investment. The first and most obvious is to study the genes, those genes and their proteins and the pathways that they are involved in that are part of the genetic form of Parkinson's disease. We know from genome studies that there are now 20, perhaps more, different genes, mutations in which cause in certain families a preponderance of Parkinson's disease. These genes, we think, are the way to study how this disease begins and progresses. It may help us, of course, to explain those particular genetic forms of the disease, but it may even help us to understand the majority of the forms of the disease that are not yet known to have a genetic basis. We want to study the neuroinflammatory process that I've described. What is it about the innate immune system in the brain that may set off the deterioration of the cells that make dopamine, or may simply make it worse? We don't even really know all the connections that a dopamine-producing nerve cell enjoys during its normal development. As I told you, nerve cells can make 1,000 different contacts with other cells. We'd like to map out, just in a normal human brain, what the circuitry is that allows a dopaminergic neuron to be produced, to be sustained, and then communicate information to other regions of the brain that control movement. And finally, there is a mysterious, sometimes decades-long period of time called the prodrome that precedes the onset of the movement disorder. And patients often experience these symptoms not knowing what they're caused by or where they will lead. Sometimes these are seemingly innocuous symptoms, such as a poorly developed sense of smell or constipation. Sometimes these symptoms can lead to Parkinson's, and patients who eventually have Parkinson's very often have these symptoms. Of course, most of the time they don't lead to Parkinson's. What is that connection? Now, another more critical early symptom that very often leads to Parkinson's is a syndrome called REM sleep disorder, where the patient experiences an inability to discriminate sleeping dreams or nightmares from actually being awake. And so these patients live out their dreams, their nightmares, while they're sleeping, sometimes flailing around in bed. They can harm themselves or their partners. And this REM sleep disorder is now being studied to see what about it leads, sometimes with 80% certainty, to the eventual development of Parkinson's disease some 10 or more years later. Well, let me just conclude by telling you where we stand in the development of this network. We have evaluated 150 teams, proposals from around the world, representing 600 different principal investigators. We've invited 70 of these teams to submit full proposals, which we're now evaluating. We will, over the next month or two, select some of these teams for funding, a really quite substantial level of funding, and we will integrate these teams into a network around the world where the teams will be expected to communicate not only within the team, but also with other teams to share their results openly and to publish their results openly as well. We hope to have a means of communication with the wider community, the patient community, but in the first instance, it's going to be an intensive, interactive, collaborative network that we hope will lead to some deeper understanding of the disease. And then once that understanding is obtained, the targets for drug development will become more clear. Well, thank you for your attention. Again, I regret I'm not able to be with you today. However, I look forward, once we're free to travel again, perhaps next year, to coming to visit Moldova, the birthplace of my maternal grandparents. Thank you for your attention.